One of the main ways in which your cell can harvest this energy is by forming ATP. ATP is considered to be the overall currency of cellular energy. However, I will note at this point that ATP is not the only form of energy in the cell. It is the most common form, but we will see as we go throughout the semester that there are other forms of energy such as GTP, TTP, CTP, etc. Remember, ATP is only a nucleotide-based energy form, and there are many different nucleotides in the cell. So ATP is very important in driving multiple different types of cellular work. This cellular work can include mechanical work, moving the cell, transport work, moving things within the cell, as well as chemical work carrying out reactions. And remember, all of these endergonic things, such as all of these reactions, must be coupled to an exergonic reaction in order to supply that energy supply to drive the endergonic processes. ATP is an important molecule that will help to couple these reactions, and it mediates most of this energy coupling within the cell. ATP is very unstable as a molecule. That means that it has a high free energy value. Whenever anything is unstable in the cell, it is usually correlated to a very high energy state. Overall, the hydrolysis or breaking down of ATP is going to release energy. Similarly, the making of ATP is going to utilize energy. So it is the making and breaking of ATP that will help to couple these reactions in the cell. This diagram here will recall your memory of what the overall structure of ATP looks like. We have an adenine nitrogenous base attached to a ribose sugar. Together, the adenine and ribose make adenosine. And then we can see that we have 3 phosphate groups attached to it. Remember that we can have AMP, adenosine monophosphate with 1 phosphate group, ADP, adenosine diphosphate with 2 phosphate groups, and here we can see ATP, adenosine with 3 phosphate groups. Let's take a look at the breakdown of ATP, or the hydrolysis of ATP. This is considered to be a very exergonic reaction, which means it's going to be spontaneous and it's going to release energy. Also, it's going to be considered a catabolic reaction. In this case, the hydrolysis of ATP, so removing a phosphate group from the ATP molecule to form ADP, has a delta G value of negative 7.3 kcal per mole. This is a very easy reaction for the cell to do, and it is also very easy to couple this with other processes. An important thing to note is here we have again the involvement of water. In this case, we're going to be adding a water molecule to the ATP in order to hydrolyze it and break it down to ADP. Also note that in this process, we have the formation of ADP, but we also have the release of an inorganic phosphate molecule as well as energy. An example of this is cellular respiration. As a whole, cellular respiration has a very negative delta G value. This means it's going to be releasing a lot of energy into the cell. This energy can be used to make lots of ATP molecules, and the formation of these ATP molecules will store the energy until the cell needs it, and so ATP will then drive other cellular work. This is an example of how ATP hydrolysis is coupled to other reactions. If you consider the first reaction at the top, this represents the overall reaction that has to be carried out within the cell. We have glutamic acid that will eventually be converted into glutamine. So we are adding glutamic acid and ammonia to form the glutamine molecule. This would be considered an anabolic reaction. We can see from the delta G value that it is 3.4 kcal per mole. So how is this energy going to be invested? Where does this energy come from? And how is this going to be carried out in the cell? So in order to carry this out, The cell will couple this reaction with ATP hydrolysis. So here we can see we've got glutamic acid. We're also going to have ATP broken down into ADP, and then we'll have the ammonia molecule added to the glutamic acid in order to form glutamine. Note that in the hydrolysis of ATP, we have the formation of ADP, but remember, it releases energy and it also releases a phosphate group. Here we can see that phosphate group is very important in tagging or being added to glutamic acid. This is an important concept that occurs in many different reactions in the cell. That inorganic phosphate release from ATP hydrolysis will go to phosphorylate a reactant or to phosphorylate or activate enzymes. So we can see the overall reaction. So even though we've got the formation of glutamine being a positive delta G reaction or endergonic anabolic reaction with a delta G value of positive 3.4, when you pair it with ATP hydrolysis, that delta G value of ATP hydrolysis alone is minus 7.3 kcal per mole. So overall, when these two reactions occur within the cell, the net delta G is going to be negative 3.9 kcal per mole. So by coupling these two reactions together, we have made an overall exergonic reaction. So by coupling these two reactions, we have now made it favorable for it to occur in the cell. This brings us to an important concept within the cell, the ATP cycle. ATP is constantly being made and broken down within the cell, dependent on what the cell needs. When the cell has extra catabolism going on, this means that there are a series of catabolic reactions which will then be releasing energy into the cell. The cell will be able to use this excess energy to make additional ATP molecules, and this will be made from joining ADP and pi, that inorganic phosphate. This will then be followed by a series of endergonic reactions. So once all of these ATP molecules are made, the cell can then utilize these ATP molecules to drive various types of anabolic reactions. And when the cell is utilizing ATP, it is hydrolyzing it and going back to forming ADP and pi. So this constant reaction of forming and breaking down ATP is very important in balancing the energetic requirements of the cell. The delta G of a reaction will tell us if a reaction is spontaneous or not, but it does not give us any indication of how fast that reaction actually occurs within the cell. So it does not give us any insight into the rate of a reaction. Some of these reactions may be spontaneous, so their delta G value may be less than 0. However, it can take a very, very long time for these reactions to actually reach an equilibrium. A catalyst can be used to increase the rate of a reaction. However, it is important to note that a catalyst will not change the delta G of that reaction, meaning that it doesn't change the amount of energy in the reactants or the product. It simply makes the reaction occur at a much faster rate. So a catalyst can be seen as a facilitator for that reaction. Catalysts are not usually consumed by the reaction, so they can be utilized multiple times to catalyze multiple reactions. When we're considering enzymes, enzymes are considered to be biological catalysts because enzymes, remember, are protein-based. Because a chemical reaction involves many complex processes such as stretching, breaking, and forming chemical bonds, we have what is termed the activation energy. This is very important because this is what prevents those spontaneous reactions from occurring within the cell in a reasonable amount of time. The activation energy creates a barrier to a spontaneous reaction occurring. This activation energy barrier is formed and is determined by the energy level in the transition state. The transition state is the point of highest G or highest energy. It is a very unstable intermediate, and remember, when you hear the term unstable, you should be thinking high energy state. So a transition state represents the highest energy state for that reaction. This is going to be very, very unstable. This is where bonds are breaking and forming at the exact same time. So enzymes can help to increase the rate of reaction simply by stabilizing that transition state. By enzymes working in stabilizing that transition state, they can lower the energy activation barrier. And remember, enzymes will not affect the delta G value. So they're not affecting the energy value of the reactants or the products. That doesn't change. They are simply lowering the energy of that unstable transition state. This is very important because this will allow for reactions, especially reactions within the cell and within the body, to reach an equilibrium state much more quickly. Thus, they increase that rate of reaction. So let's take a look at what that looks like in a reaction profile. This diagram here represents the energy profile of a reaction. Once again, we have the free energy value, or G value, present on the y-axis, and we have the progress of the reaction on the x-axis. Notice that in our reactants, we have molecule or atom A joined with B, atom C joined with D. Take a look at what we want to form in our products. In our products, we want A bonded to C and B bonded to D. Overall, our products have a much lower energy state than our reactants. So overall, the delta G value of this reaction is going to be negative, less than 0. This would be considered an exergonic reaction. Energy is going to be released, and this is spontaneous. However, even though this reaction is spontaneous, let's take a look at how this reaction would actually proceed in the cell. Here we have the transition state. Remember, the transition state represents the highest unstable point in a reaction where bonds are going to be broken and formed at the same time. So here we can see, representative of the squiggly line, in this transition state, we are going to be breaking the bond between A and B and breaking the bond between C and D, and at the same time forming the bond in our products. So we're trying to form the bonds between A and C as well as B and D. So this is considered very, very unstable. And here we have, at that energy value, we can see that it is the highest energy state of this reaction, and this is what creates that EA, or energy activation barrier. So now let's take a look at what happens when we have the presence of an enzyme. So this diagram represents the energy profile, similar to what we've seen before, and that would be represented by the black line. So we can see that the energy activation barrier without an enzyme is very high. This means that even though the reaction is spontaneous, you still need to be able to invest energy into this reaction in order for it to occur, specifically the amount of energy that would be referred to by the EA, or activation barrier. Now, when we have the presence of an enzyme represented in the red line, we can see that the enzyme acts to stabilize that transition state. It would lower the energy in that transition state, and thus it would lower the activation energy barrier. So this would make it much easier for this reaction to occur in the cell and increase the rate of that reaction. Keep in mind that the energy state in the reactants and energy state of the products are not going to be changed by the presence of an enzyme. The only thing that changes is the energy activation barrier, or EA. We can still see that the overall delta G is going to be similar and is unaffected by an enzyme. So catalysis is a very important process in the cell. This is what the cell uses in order to get all of these reactions that would normally not occur within the cell to occur by using enzymes as catalysts. They will facilitate these complex chemical reactions. How they are facilitated is by stabilizing the transition state. of the reaction. Because we're working with an enzyme, we now have to be concerned with the substrate as well as the active site. So when we have an enzyme present in the reaction, we can refer to the reactants as substrate molecules. These are the initial molecules that are going to be acted on by the enzyme. We also can refer to the active site. This is going to be the region on the enzyme that binds to the substrate and where that transition state is going to be stabilized. This will be determined by the amino acid side chains that are important in forming that active site. Remember that now we have a complex enzyme structure. We're going to have hydrogen bonds, electrostatic attractions, van der Waals forces, etc., that are going to be very important in holding those substrate molecules to the active site. In addition to stabilizing the transition state, enzymes may participate in the reaction. This means that an enzyme can facilitate thousands of reactions per second. They can be used multiple times over and over again while not changing the overall shape and function of the enzyme. This diagram here represents enzyme action within the cell. So we start with our substrate molecules in red and green. They're going to be present within the cell and they are going to be recognized by the enzyme. They're going to bond to the active site and form an enzyme substrate complex. This complex is going to be held into place by those weak interactions we mentioned before, van der Waals forces, hydrogen bonds, etc. Remember that the enzyme works in lowering the transition state energy level and thus lowering the activation energy barrier. So the reaction is going to occur much faster with the presence of an enzyme. These substrate molecules are going to be converted into your product molecules. However, an enzyme, remember, is a protein, so it's going to be very specific with respect to the active site and what it can bond with. So once those products are formed, they no longer fit into the active site properly. They are going to then be released by the enzyme into the cell. The enzyme will then take on its original conformation and thus be open to catalyze more reactions in the cell. Enzymes sometimes require a little bit of help to become active within the cell. This will help to control their activity. So in this case, we would have the involvement of either cofactors or coenzymes to assist the activity of the enzyme. If the molecule that is assisting is non-protein based, we term that a cofactor. This would be very common in your metals, such as zinc. If you have an organic molecule that facilitates or activates the enzyme, we would then call that a coenzyme. Most of the vitamins, vitamin A, vitamin K, for instance, are coenzymes for important reactions in the cell. Another way to control enzyme activity is by having optimal windows. And remember, we've already seen this concept with the action of a lysosome, where there are many hydrolytic enzymes within the cell that are only active at a lower pH, and that lower pH is only present within the lysosome. That is an example of optimal enzyme activity. So this could be with respect to pH or also with respect to temperature. This diagram here helps to illustrate the concept of optimal enzyme activity, both with respect to temperature or with respect to pH. On the top of this graph, we can see optimal temperature for two different enzymes, one being a typical human enzyme or enzyme found in the human body. The optimal temperature for their activity is 37 degrees, which also corresponds to typical body temperature. However, if we take a look at the optimal window of activity for temperature in a thermophilic or heat-loving bacteria, such as sulfur bacteria that is common in the sulfur springs, we can see that the optimal temperature for that type of bacteria is 75 degrees centigrade. So this optimal range is very different than what we would find in enzymes in the human body. We can also have that same concept apply for pH. If we take a look at the optimal pH range of pepsin, which is a stomach enzyme, we can see that the optimal pH peaks at around pH of 2, and that corresponds to the typical pH of an active stomach. However, We have trypsin, which is also a human enzyme. However, this is an enzyme that is active in the intestinal tissue of cells and organisms. We can see that the pH is much higher, a pH of 8. And this also corresponds to the typical environment that is found within the intestinal area, as opposed to the stomach. Another way to control enzymes and their action within the cell is by having inhibitor molecules. Enzyme activity can be impaired by inhibitors. This can be irreversible, meaning that it can cause permanent changes in the enzyme, or it can be reversible, meaning that there are weak bonds that hold that inhibitor in place, and that inhibitor can easily leave the enzyme for the enzyme to then become active. We can divide these inhibitors into competitive or non-competitive inhibitors. Competitive inhibition uses a competitive inhibitor. This is going to be important because the inhibitor molecule is going to bind at the active site. Remember, enzymes are very specific. So if this inhibitor molecule is binding to the active site, this means that it would also resemble or mimic the substrate molecule. This is considered to be direct interference. The competitive inhibitor molecule is going to be directly competing with the substrate molecule for access to the active site. However, a non-competitive inhibitor will still bind to the enzyme, but it binds to an allosteric site, not to the active site. As a result, the inhibitor molecule is not going to look like the substrate molecule. We can also consider these non-competitive inhibitors to be allosteric regulators. This is because they bind to the allosteric site on the enzyme, not the active site. How these inhibitors work is indirectly because they bind to the allosteric site and change the conformation of the enzyme to then change the active site so that it no longer binds to the substrate molecule. This is considered to be indirect, whereas competitive inhibition, remember, is direct interference. Another way to control enzymes is by looking at the overall pathway. So these pathways, metabolic pathways, can be controlled by not being turned on all of the time. So these pathways can be turned on and turned off depending on what the cell or what the organism needs. Sometimes enzymes are only going to be made when needed, so that means that certain areas of the DNA will be transcribed and translated at different times in order to only make these enzymes when they're needed, which makes sense because making an enzyme is utilizing energy. So why are you going to use your energy to make enzymes when they're not needed in the cell? Also, you can control metabolic pathways but through allosteric regulation. And this overall control of the pathway can be positive or negative, meaning that it can be used to turn on pathways, but you can also control it by shutting down these pathways. And how this is done is through the concept of feedback inhibition. This is one of the most common ways to control a metabolic pathway. This is when the final product of a pathway, when it's being produced in a high concentration, will actually turn around and inhibit the enzymes involved in producing that product. This diagram here helps to illustrate that metabolic control in the cell. In the first diagram here to the left, we have the role of allosteric activators and inhibitors on an enzyme. Let's take a look at the first form of the enzyme, which is the active form. There are two different sites on the enzyme. This enzyme here is made up of 4 subunits. Each subunit has an active site, which would be used to catalyze the reaction and stabilize the transition state. We also have the regulatory site or allosteric site. In the top part of the diagram, we have an activator molecule in green that comes in to bind to a regulatory site. This will then stabilize the active form of the enzyme, meaning that it will be locked into the active state. It will then go on to perform its function in the cell. However, the active form of the enzyme can oscillate between the active and inactive form. If we take a look at the inactive form, here we've got a non-functional active site. Notice that the conformation of the active site is different than it is in the active form. Enzymes are very specific, so when you have a simple change in conformation of the active site, it's no longer going to be able to bind to the substrate molecules. In the inactive form, in this case, in the lower part of the diagram, we have a red inhibitor molecule that comes in, binds to the regulatory site, and in this case, it will stabilize the inactive form and effectively shut down the enzyme. In diagram part B of this image, we have another type of allosteric activation where we can have the inactive form of an enzyme binding to the substrate molecule and becoming stabilized in the active form. This will then go on to perform its function in the cell. This diagram here illustrates that concept of feedback inhibition. In this case, we're going to have the final product of the pathway come around and shut down the enzymes involved in the pathway. So we can see from this diagram, we have an enzyme that has its active site available. It is going to bind to the substrate molecule and then produce a final product. In this case, it is through a series of different enzymes and a series of forming different intermediate compounds to finally form the end product in this case, which is isoleucine. In feedback inhibition, we have the final product coming back to shut down the enzyme. So you can see here that the end product, isoleucine, is going to come around and bind to that enzyme. It is going to bind to the allosteric site and regulate that enzyme. It will shut it down by changing that active site. In essence, this will shut down that pathway. The isoleucine concentration will eventually lower, and when it reaches a much lower level, the isoleucine will then be removed from the enzyme. This pathway would then be activated once again, and then you would see isoleucine concentrations in the cell increase. So there's many different ways that enzymes can be controlled. They can be controlled directly, indirectly through inhibition, and they can also be controlled via pathway mechanisms such as feedback inhibition. And that concludes the introduction to metabolism and enzyme action in the cell.